Hi, everyone. Today we're going to talk about chapter 7, which is about the lymphatic and immune systems, which some people consider one system, some people separate it and consider it two different systems. Either way, we put them together in the same chapter. So our lymphatic system has its own lymphatic vessels and organs. The main functions that contribute to homeostasis are lymphatic capillaries will absorb excess interstitial fluid and then return it to the bloodstream. Lymphatic capillaries also absorb fats from the digestive tract and transport them into the bloodstream, and then lymphoid organs help defend the body against disease. This whole chapter, we will be defending against disease. This function is mainly carried out by the white blood cells present in the lymphatic vessels, lymphoid organs, and the bloodstream. Lymphatic vessels carry a fluid called lymph, so blood is not the only liquid we have inside of our bodies, just like arteries and capillaries are not the only vessels we have. We have lymph vessels, carry the fluid lymph. It begins as lymphatic capillaries in the tissue. As lymph moves away from the tissues, the capillaries merge into lymphatic vessels and then to two lymphatic ducts, the thoracic duct and the right lymphatic duct. The larger thoracic duct collects lymph from the body below the thorax, the left arm, and the left side of the head and neck, and empties into the left subclavian vein. The right lymphatic duct returns lymph from the right arm and the right side of the head and neck into the right subclavian vein. So here's just a brief diagram of the lymphatic ducts and organs and tissues in our upper body. There's the lymph coming from the right side goes to the right subclavian vein, from the left side goes to the left. The anatomy of larger lymphatic vessels is simpler than that of cardiovascular veins, including the presence of valves, similar to. Valves prevent lymph from flowing backward, just like it does in our blood vessels. The movement of lymph is largely dependent on skeletal muscle contraction. Every time we flex our muscles or move, it moves our lymph inside of us. There's two types of lymphatic organs, primary and secondary. The primary lymphatic organs are red bone marrow and the thymus. The secondary lymphatic organs are lymph nodes, the spleen, and the tonsils. We'll talk about the differences between all that. Red bone marrow is the site of blood cell production. In children, most bones have red marrow, only a few do in adults. And B cells or B lymphocytes mature here. Those are found in the end of our long bones, like in the picture. That's a picture of our femur, which is one of our long bones. That's where the red marrow is found. And then the yellow marrow is found in the hollow cavity, the actual long part. The thymus is bilobed, means it just has two lobes. Looks in the thoracic cavity superior to the heart. It is the largest in children, and it actually shrinks as we age. Its two main functions, one is to produce thymosin, which is a hormone that induces the maturation of T cells or T lymphocytes. Immature T cells move from the marrow to the thymus, where they mature before they move on and complete their job. The spleen filters blood. The upper left region of the abdominal cavity, kind of right behind the stomach. Connective tissue divides into white pulp and red pulp. Macrophages in red pulp remove pathogens, debris, and worn out red blood cells from the blood. It has a thin outer capsule, so it can rupture from trauma. The spleen is actually very, very delicate, and it ruptures if somebody's in an accident or even gets hit too hard, the spleen can rupture. Lymph nodes are found along the lymphatic vessels. They filter the lymph. Connective tissue forms a capsule around it and divides it into compartments. You can see in the top right. Let me move myself. That's the inside of a lymph node. You can see it looks like a tiny little kidney, but it has its own little compartments inside. It's filled with macrophages that engulf pathogens and debris. It also houses lymphocytes, which fight infections and cancer cells. It is named for their location. They're very common in the neck, armpit, and groin. The axillary nodes in the armpits, inguinal nodes are in the groin. That's why when you get sick, your doctor will feel along your neck right here. Because when you're sick, your lymph nodes will swell and you have a collection of them right here. So it's easy to tell if your lymph nodes are swollen or not. Other secondary lymphatic organs, the lymphatic nodules are concentrations of lymphoid tissue that don't have a capsule. Tonsils are located in the pharynx. They have the same function as the lymph nodes. They fight infections that come through the nose and the mouth. Peyer's patches are found in the intestinal walls and the appendix. They fight infections that come in via the digestive tract. Anything you eat has bacteria or viral matter on it. Parasites, your digestive tract will try to fight it off before it gets into your blood. Immune defenses are next. Innate immune defenses are next. So immunity, killing or removing of foreign substances, pathogens, and cancer cells from the body. There are two branches of our immune system, the innate immune system and the adaptive immune system. The innate is fully functional without previous exposure to a pathogen, and the adaptive is initiated when exposed to a pathogen, either naturally or via something like a vaccine. Innate immune defenses include physical and chemical barriers. If it is breached, it will lead to an inflammatory response. Innate defenses have no recognition of a pathogen and no memory. There are things like our skin that will just completely keep everything out. That is its job. Doesn't matter what it is. The physical barriers, they're our first line of defense against pathogens, our skin and our mucous membranes. The skin has a lot of keratin, so it's tough. Also, exfoliation carries microbes away from the body. We all should be exfoliating. And then mucous membranes have ciliated cells. The cilia sweep away mucus with entrapped pathogens. We have those throughout our nose and our mouth and all the rest of our openings. Chemical barriers are acidic secretions of the sebaceous glands. They will weaken or kill bacteria on the skin. Sweat, saliva, and tears have lysozyme, which is an antibacterial enzyme. The acidic pH of the stomach will inhibit or kill bacteria. Our stomach has a pH of, I think, maybe between 2 and 3. Most bacteria don't like it very much. They'll die. Normal flora, which are microbes that usually reside in the mouth and the intestine, prevent potential pathogens from taking up residence. And chronic use of antibiotics can make one susceptible to infection by killing off our normal flora. So when you take antibiotics, you tend to get some infections because it kills off what's normally there. We have normal flora, normal bacteria that live inside of us and help keep our pH balance, help us digest food. That's completely normal. But when we take antibiotics, it'll kill the bad bacteria and it'll kill our own bacteria as well. There's a short video about the inflammatory response.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria. The inflammatory
Protective proteins. The complement system, several plasma proteins designated by the letter C and a number. They're involved in and amplify the inflammatory response. Some bind to mast cells, triggering the histamine release. Others attract phagocytes to the scene. Some form a membrane attack complex that produces holes in the surface of bacteria. Fluids enter the bacterial cells and then they burst. Interferons are chemicals produced by cells that are infected with viruses as a warning to other cells. They bind to receptors of non-infected cells, causing them to produce substances that will interfere with viral replication. There's a little mini diagram of that happening, signals neighboring uninfected cells to destroy RNA and reduce protein synthesis. It will signal neighboring cells to undergo apoptosis, which is just programmed cell death. I'm not healthy, so I'm going to kill myself before I infect other cells, and it will activate immune cells. And then adaptive immune defenses. Adaptive defenses come into play when innate or the nonspecific defenses, like the skin and the chemicals, have failed to prevent an infection. They overcome an infection by doing away with the particular disease-causing agent that has entered the body. They establish a mechanism for the immune system to remember the action. They provide some protection against cancer. How they work. They respond to antigens, which are pieces of pathogens that our immune system recognizes as foreign. Fragments of bacteria, viruses, molds, and parasitic worms can all be antigenic. They all have little antigen pieces on them that our body will say, nope, you don't belong here. Abnormal plasma membrane proteins produced by cancer cells may also be antigens. The immune system is able to distinguish self, which is our own cells, from non-self, the pathogens with the antigens on them. Adaptive defenses depend on the action of B cells or T cells, B lymphocytes or T lymphocytes. Each have a specific antigen receptor that binds to specific antigens. Each lymphocyte has only one type of receptor. The receptor and antigen fit together like a lock and a key. There are two pathways of adaptive immunity, cell-mediated and antibody-mediated. In cell-mediated immunity, T cells will kill any other cells that are presenting the specific foreign antigen. In antibody-mediated immunity, also called humoral immunity, B cells produce antibodies that will bind to the free antigens in body fluids. B cell receptors are protein receptors found on the surface of B cells. An antigen will bind to the BCR on only one type of B cell. That one B cell will then clone itself. That is called clonal expansion. And clonal expansion produces antibody-secreting plasma cells as well as memory B cells. They are very, very useful. The fate of activated B cells.
B cells play a key role in our immune system's ability to recognize and fight specific pathogens. In this video, we're going to break down the processes of B cell activation, antibody production, and memory cell formation, all a part of the humoral response. So let's do it. Hey, Leslie Samuel here from Interactive Biology, where we're making biology fun. And I want you to imagine something with me. You're taking a walk in the park, enjoying a beautiful day. The sun is shining, everything seems perfect. All of a sudden, you feel a sharp sting on your arm. You smack your arm, and then you notice that you killed a mosquito. But it's not just any mosquito. You've just been bitten by a mosquito carrying the West Nile virus. Now, you don't know this, so you just go about your business. But even though you don't fully understand what just happened, your immune system does, and it springs into action. And B cells are at the forefront of this battle. Let's look at this more closely. B cells are immune system cells that recognize antigens and trigger a series of immune system actions. When a mature B cell encounters a pathogen, something like the West Nile virus, there are B cell receptors on its surface that recognize and bind to a specific antigen. I'll call these B cell receptors BCRs from now on. The B cell then takes in the antigen BCR complex and encloses it in a vesicle. And inside the vesicle, the antigen gets broken down into smaller pieces called peptides. The B cell then presents these peptides on its surface using molecules called MHC, class 2 molecules. Now the B cell is acting like an antigen presenting cell, or APC. It's presenting the antigen on the surface of the cell. The name makes sense. The next thing that happens is that helper T cells come into play. T cell receptors, I'll call them TCRs, they recognize and bind to the West Nile virus antigen MHC2 complex on the B cell surface. And this interaction, along with additional signals from the helper T cell, things like CD40L binding to CD40, provide the necessary co-stimulation for the B cells to become fully activated. Now that we have a fully activated B cell, something interesting is going to happen. It's going to go through a process called clonal expansion, meaning it rapidly divides and differentiates into two main types of cells: plasma cells and memory B cells. Plasma cells are like antibody producing factories. They secrete a bunch of antibodies, and these antibodies are like the heroes of the immune system. They're specialized proteins designed to recognize and neutralize specific antigens, such as those on the West Nile virus. These antibodies are released into the bloodstream, where they can do one of three things. Number one, they can bind to and neutralize the virus, which is a good thing. Or number two, they can trigger the complement system. This is also a good thing because it helps to fight against harmful pathogens. I go into a lot of detail about the complement system in another video, and you can check that out to learn more. Or number three, they can mark it for destruction by other immune cells, such as phagocytes. And these phagocytes, they'll just come into the area and start eating stuff up. Pretty cool. This entire process is known as the humoral response, and it's instrumental for keeping you safe from the West Nile virus and many other pathogens. Now, memory B cells serve a different purpose. They don't produce antibodies immediately. Instead, what they do is they provide long-lasting immunity by remembering that specific West Nile virus antigen that they encountered. What happens is, if the same virus re-enters your body in the future, these memory B cells quickly recognize it, rapidly multiply, and differentiate into plasma cells and more memory B cells. Now, this secondary immune response is even faster, it's more potent, because the memory cells are just there sitting and waiting for that specific antigen. It's like they're a special ops agent just waiting in the background for that one invader to come in. And when it does come in, they stop it dead in its tracks. This helps to prevent reinfection by the West Nile virus. During the humoral response, some activated B cells also undergo processes called isotype switching and affinity maturation. You see, there are different types or isotypes of antibodies. There's IgG, IgM, IgD, and IgE. These different isotypes differ in their distribution and also their functions. Isotype switching involves changing the class of antibody produced, for example, from IgM to IgG, in order for them to better target the pathogen that the body is trying to get rid of, in this case, the West Nile virus. And I also said that they go through a process of affinity maturation. Now, this is a process that helps to refine the antibody's ability to bind more tightly to the virus's antigens. It's basically increasing how effective it is in neutralizing the virus. And that's it. That's my guide to B cell activation, antibody production, and memory cell formation. As you can see, the humoral response plays a vital role in the function of the immune system. But there's also the cell-mediated response that plays a crucial role in the function of the immune system. And I'll cover that in the next video. My name is Jesse Samuel from Interactive Biology, where we're making biology fun. That's it for this video, and I'll see you in the next one. B cell.
So apoptosis, I mentioned it a little bit ago. But after an infection has been overcome, the plasma cells are no longer needed. They undergo a process called apoptosis, which is just programmed cell death. They commit suicide so as not to infect anything around them. The structure of an antibody, we saw it a little bit in the video. It's a Y-shaped protein with two arms. Each arm has a heavy or a long polypeptide chain and a light or a short polypeptide chain. These chains have constant regions located at the trunk of the Y, where the sequence of amino acids is fixed. Antibodies are classified by the structure of its constant region. The variable regions form an antigen binding site, which the shape is specific to the particular antigen. The antigen combines with the antibody at the antigen binding site in a lock and key manner. Antibodies coat viruses and toxins completely while neutralizing them and removing the threat. the immune complex, which is the clump of antigens combined with the antibodies. And the immune complex will attract the white blood cells to come and eat them. There are 5 classes of circulating antibodies: IgG, M, A, D, and E. IgG is the major type in the blood. Smaller numbers are found in the lymph and the interstitial fluid. They bind to pathogens and toxins. They will cross the placenta from the mother to the fetus, which confers temporary immune protection. IgM are pentameters, meaning they have 5 sides. These are the first antibodies produced by a newborn, the first to appear in the blood after an infection begins, and the first to disappear when the infection is over. These will also activate the complement system. IgA are monomers or dimers containing 2 Y-shaped structures. It's the main type of antibody found in body secretions, saliva, tears, mucus, breast milk. They bind to pathogens, so they can't reach the bloodstream. IgD is an antigen receptor on immature B cells. IgE prevents parasitic worm infections and can also cause allergic responses when needed, when our bodies run into something we're allergic to. Monoclonal antibodies are all identical and produced by plasma cells. They can be produced in a lab. They are used for medical tests and treatments. Now we are on to T cells, produced in the bone marrow, mature in the thymus. T cell receptors are unique receptors on the surface of each T cell. Unlike B cells, T cells are unable to recognize an antigen without help. It must be displayed to them as an antigen-presenting cell. That's what the B cells do. They ingest the antigen, they take a piece of it, and then they display it on their surface, so then the T cell can recognize it. After phagocytizing a pathogen, APCs travel to the T cells in a lymph node or the spleen. APCs break the ingested pathogen apart in a lysosome. A piece of the pathogen is then displayed on the groove of a major histocompatibility complex, or MHC protein, on the cell's surface. There's 2 classes, MHC1 and 2. So you can kind of see in the GIF there. And then a pathogen will get into a cell. The cell will break it down in parts, and then it will present part of it on its surface. Human MHC2 proteins are called human leukocyte antigens, or HLAs. They are found on all body cells. There's 3 general groups, HLA, B, and C. I'm sorry, HLA-A, B, and DR. Each person has a unique combination of HLAs. HLAs need to be well matched for organ transplants. That is what doctors look at, the pieces that need to match. HLAs of identical twins are identical, which is why if you ever need an organ, an identical twin or a close sibling is your best match. And then T cells also go through clonal expansion. Many copies of the activated T cell are produced. It occurs when the APC presents the antigen to the T cell that has the specific T cell receptor that will bind to the particular antigen. This activates the T cell, causing it to undergo clonal expansion. Some T cells will become cytotoxic or killer T cells, and others will become helper T cells. And hopefully you can see in the GIF down there, you have the antigen from the APC connects with that T cell receptor, or TCR. Cytotoxic or killer T cells have storage vacuoles that contain perforins and enzymes called granzymes. After binding to a virus-infected cell or a tumor cell, it releases these perforins, which will punch holes in the plasma membrane and form a pore. Cytotoxic T cells then deliver granzymes into the pore, causing the cell, the infected cell, to undergo apoptosis. Helper T cells secrete cytokines that enhance the response of all types of immune cells. B cells cannot be activated without T cell help.
Most cells that display CD4 become helper T cells. CD4 T cells can only recognize foreign antigens when they are presented in conjunction with major histocompatibility complex, or MHC class II molecules. These protein molecules are only found on certain immune system cells, called antigen-presenting cells. The helper T cell antigen receptor must match the presented MHC2 antigen. Their binding with one another is the first signal needed to begin activation of the T cell. A second signal, known as co-stimulation, must also occur. Pairs of molecules attached to the membranes of the T cell and the APC cell help bind the cells tightly together and can provide co-stimulation. Cytokines, small protein hormones, are released by the antigen-presenting cell and are co-stimulators. Once the T cell is activated, it proliferates into clones of itself. Most of these new cells differentiate into effector T cells. The receptors on these cells are able to bind to the same antigen as the receptor on the original T cell. Some of the T cell offspring will not become effector cells, but remain as memory T cells, which will respond rapidly upon future exposure to the same antigen. Once activated, helper T cells secrete cytokines. One cytokine is interleukin-2, which is important in most immune responses. It is also known as T cell growth factor, since it acts as a co-stimulator for T cells. It also enhances the activation of B cells and natural killer cells. Other cytokines are interleukin-4, or B cell stimulating factor, and interleukin-5, which is also involved in B cell co-stimulation. In addition, helper T cells can secrete gamma interferon, which stimulates phagocytosis and nonspecific resistance. Gamma interferon is involved in both cell-mediated and antibody-mediated immunity. In general, individual helper T cells do not secrete all cytokines. Subsets of helper T cells secrete different cytokines. The subset of helper T cell that is activated is determined by which defense is needed against a specific pathogen, and will also determine which cytokine is secreted.
Next section is acquired immunity. So immunity can be brought about naturally through an infection or artificially through medical intervention. Acquired or adaptive immunity can be classified in several ways. So we have immunity, we split that into adaptive and innate. Now we're splitting the adaptive into more categories, and we will go through all of these. So a person's body makes antibodies against a particular antigen. We just went through all that. It can happen through natural infections or immunizations involving vaccines. These contain antigens from the pathogen or pathogens themselves. They are treated so they can no longer cause disease. The first exposure to an antigen produces a primary response. The second exposure, a secondary response. And this whole process depends on memory lymphocytes, like the memory B cells. Also, sometimes booster shots are needed, because after a while, our body does forget. Passive immunity. An individual is given antibodies to combat a disease since it is not produced by the individual's plasma cells. Passive immunity is temporary. Newborn infants are passively immune to some diseases because those IgG antibodies have crossed the placenta from the mother's blood into the child's blood. Breastfeeding prolongs this natural passive immunity because IgG and IgA antibodies are present in mother's breast milk. Humans can also receive antibodies by injection. Cytokines are signaling molecules produced by T cells, macrophages, and other cells. They regulate white blood cell formation and function. Interferons and interleukins are cytokines. They are used in various medical treatments. And the video we just watched went through all the different kind of interleukins and the gamma interferon. And our last section is about disorders of the immune system. HIV and AIDS is a huge one. The human immunodeficiency virus, or HIV, the virus causes acquired immunodeficiency syndrome, or AIDS. There are two main types of HIV. HIV-1 is more widespread. It is also more virulent or more infectious. There's also HIV-2. HIV-2, found in the green monkey, lives in Western Africa. HIV-1 replica virus found in a subgroup of chimpanzees, that was once common in West Central Africa. This suggests HIV viruses originated in non-human primates, then mutated to HIV after humans ate the non-human primate meat. That is the best theory right now on how HIV jumped from primates like monkeys into humans. HIV. Helper T cells have protein receptors called CD4 and CCR5 on their surface. They are like locks that keep most things out. These are called co-receptors. HIV has to bond to both of them to get into the cell. Once the T cells are destroyed, HIV gets into the bloodstream and will infect the surrounding T cells, specifically the surrounding helper T cells. The original HIV infection becomes AIDS when the number of T cells falls below 200 cells per microliter of blood. Normally, we have 5,000 to 10,000 T cells per microliter of blood. Microliter is a very, very tiny drop. But once HIV has killed most of them, it becomes AIDS. And then opportunistic infections. Pathogens take advantage of the low T cell count and destroy them faster than we can make new ones. People don't actually die from HIV or AIDS. They die from the infections that take over their weakened immune systems. Often it's either pneumonia, cancer, or a bacterial infection. Transmission and prevention. Sexual contact with a person with HIV is the most common, so condoms are the most effective prevention. Needle sharing among people who use IV drugs. There are syringe service programs that many, many homeless people use in large cities that have lowered the rate of HIV transmission in those cities. Transfusions of infected blood or blood clotting factors, they're very rare today. But it used to be in the 80s a link between hemophilia and AIDS before people realized how both of those diseases worked. And then babies born to women with HIV before or during birth, or even through breastfeeding. The one body fluid it's not found in is saliva. So kissing doesn't do it. Body fluids with the highest concentrations are blood, semen, vaginal fluid, and breast milk. HIV is not transmitted through contact in the workplace, schools, social settings, kissing, hugging, shaking hands, touching toilet seats, doorknobs, dishes, drinking glasses, food, pets, or mosquitoes. Mosquitoes is a big one that persists for some reason, but no, HIV is not transmitted through mosquitoes. There is no cure for HIV/AIDS yet, and it can take up to 6 months to show up in tests. Individuals immediately undergo antiviral treatment following a positive HIV test result. Highly active antiretroviral therapy, or HAART, usually can stop HIV replication, where the viral load will become undetectable. HAART uses a combination of drugs that interfere with the life cycle of HIV. When HAART is discontinued, the virus will rebound and just start replicating itself and start infecting cells all over again. So it's a lifetime thing. Other treatment options, possible drug combinations, entry inhibitors to stop HIV from entering a cell. The virus is prevented from binding to CD4 or CCR5. Reverse transcriptase inhibitors, such as zidovudine, also known as AZT, interfere with the operation of the reverse transcriptase enzyme, because HIV uses reverse transcriptase once it gets into a cell. Integrase inhibitors prevent HIV from inserting its genetic material into that of the host cells. Protease inhibitors prevent protease from cutting up newly created polypeptides. Assembly and budding inhibitors are in the experimental stage, and none are available as of yet. But you can see we've been trying since the late 80s, early 90s to come up with drugs to combat HIV. Over 21 million people worldwide are now receiving HIV treatment. There are some drug therapy dangers. People with HIV may become lax in their efforts to avoid infections, knowing that drug therapy is available. Drug use leads to drug-resistant viruses, so some HIV viruses have become drug-resistant when patients fail to adhere to their drug regimens. People with HIV have to take their medication as prescribed every single day at the same time to keep that viral load low, or else it risks coming back. There's also hypersensitivity reactions, when the immune system overreacts and causes harm to the body, like allergies, receiving an incompatible blood type, tissue rejection, and autoimmune diseases. We're moving on from HIV, by the way. These are other things that can happen to a person's immune system. Hypersensitivity to allergens, such as pollen, food, animal hair, which would normally be harmless. Those are what? Just the basic definition of allergies. An immediate allergic response can occur within seconds of contact with the antigen. It is caused by IgE antibodies attached to receptors on mast cells and basophils, which will release histamine when they bind these allergens. When pollen is an allergen, histamine stimulates the mucous membranes of the nose and the eyes to release fluid. Runny nose, watery eyes, hay fever. With asthma, the airways also constrict, resulting in wheezing. That person has a hard time breathing because their airways get smaller and smaller. Food allergies can cause nausea, vomiting, diarrhea, and even death. You know anybody with a peanut allergy? Some peanut allergies are very, very serious. Anaphylactic shock is an immediate allergic response. It occurs when the allergen enters the bloodstream. It is characterized by sudden and life-threatening drop in blood pressure due to increased permeability of the capillaries by histamine. Injecting epinephrine or adrenaline can counteract this reaction. Delayed allergic responses are initiated by memory T cells at the location of contact with the allergen. Transplant rejection. The recipient's immune system recognizes the transplanted tissue is not self. It is other. Cytotoxic T cells attack the transplanted tissue, but this can be controlled with immunosuppressive drugs and by transplanted organs that have the same MHC proteins in the donor and the recipient. Some immunosuppressive drugs act by inhibiting the production of cytokines. Xenotransplantation is the idea that we can transplant organs from an animal. This is already happening. And some organs can be grown in a lab now with stem cells. SCID stands for Severe Combined Immunodeficiency Disease. Both antibody and cell-mediated immunity are inadequate or lacking. A minor infection can be fatal. Treated with bone marrow transplant or gene therapy, one of the longest known survivors of this condition was David, known as the boy in the bubble, who died at 12 years old after being isolated in a sterile plastic bubble for all but the last 15 days of his life.
Being isolated
That's 8 minutes long. I will link that for you guys if you want to watch it.
at home,
And then autoimmune diseases. Cytotoxic T cells or antibodies attack the body's own cells as if they were foreign. It involves both genetic and environmental factors, sometimes follows an infection. For example, rheumatic fever. Antibodies induced by a streptococcal bacterial infection of the throat also react with heart muscle that damages the heart muscle and the heart valves. Rheumatoid arthritis, when our antibodies react against our joints. They don't like our joint tissue, so they start attacking them and give us arthritis. Lupus, officially called systemic lupus erythematosus. There's various symptoms: facial rash, fever, joint pain. It causes damage to the central nervous system, heart, and kidneys, and it can be fatal. They produce high levels of anti-DNA antibodies. All humans, except for red blood cells, contain DNA, so the symptoms of lupus can be exhibited throughout the body, wherever our cells have nuclei. Myasthenia gravis. Antibodies attach to and interfere with the function of muscles. The result is severe muscle weakness, eventually resulting in death from respiratory failure, because we actually need our muscles in our thorax to function properly so we can breathe properly. And multiple sclerosis. Our T cells will attack the myelin sheath covering our nerve fibers, causing central nervous system dysfunction, double vision, and muscular weakness. MS might not be an autoimmune disease because a specific antigen has not been identified yet, but treatments for all of these diseases are drugs designed to decrease immune response, because the longer an immune response goes on, the more it's actually going to harm our bodies. And that is it for chapter 7.